analysis and discuss about some statistical method. Basically, it's two statistical method, GSEA and Fisher test. So basically, what is the gene set enrichment analysis? So it is a method to identify the class of proteins that are overrepresented in a largest set of genes proteome, and they have a well-proof association with any disease phenotype. So suppose like some set of genes is involved in some specific function, that means that proteins are affected or involved in particular pathway that affect the that are altered in the disease condition. So that's why gene set enrichment analysis is important for us. Yeah. So there is a some file format how it looks like here in the graphic representation, it is showing that any process pathway has some specific group of proteins that perform some specific function. So we have to identify that specific proteins function in with respect to gene set enrichment analysis. So this is the output file. Suppose some metabolism, and I am taking like one first example. Like we have like in metabolism, like how its important significance. So they have p values, and like we have 1621 proteins as a query protein, and out of this, these 323 proteins are involved in metabolism. As similarly in other pathways, these proteins are involved, and these are the experimental evidence. So that's why it is very important for us to know the in our query protein what are the proteins which are most affected during disease condition. These are the two methods like GSEA, as I earlier said, and Fisher exact test. So in a GSEA file format, you need a list of genes and their properties, like some weighted, like a fold chain. It could be fold chain, p-value, and other entities which can define the weightage of particular gene or rank. But in the Fisher exact test, only we have need only list of proteins genes for the enrichment. So these are the some method tool, online tool, which can be which can give the gene set enrichment result by using this. So these are the our like protein intensity file where we can just take a average of wild type versus disease type, and then take a average and calculate the chain, and then take the gene name and fold chain and use as a query for query protein list for input file for a GCA, as well as we can use also for Fisher test from this protein. So how this method works? So as I said, like we have two classes of two groups we need for making a matrix. Like first is like a wild type and second is disease. So suppose sample 1, sample 2, sample 3 is came in the wild type, and sample 3, 4 and so on is in that and disease 2. So when like we calculated all these fold chain, log fold chain and t-test, so make a rank and order the protein in the list as by rank. Like suppose higher fold chain to lower fold chain, yeah, lower p-values to less p-values, and just go for GCA. So here is an option like we can provide a GMT file. Basically, it is a GMT file. It is a like gene matrix table, which is like predefined of already a matrix of each whole proteome. So like suppose one function is defined and how it looks like suppose uh, I said like one function proteome like a metabolism. So then in the metabolism, how number of proteins, among 20,000 proteins are involved in the metabolism. So these are the detailed file actually. So we can provide in the input list, as well as we can just go by using the package like Reactome RA package in R, we can use directly. So I prefer mostly like package, because in GMT file you have to be updated every day because many entries are added, some deleted. So suppose like in the if you're taking like Reactome package, there is a benefit that these packages are updated like frequently, so and easy to use. So these GMT files, I will tell you how I can download these GMT files from the molecular signature databases. So, and similarly, like Fisher exact test, this is the simple permutation combination formula, which is shown. Like I am focusing here. Like suppose in apoptosis, like one protein involved and 13 proteins are not involved in out of 14 query protein. So here you have to remember that these are the query protein, and these are the protein interactome protein. So these are the interactome proteins which are totally like out of 20,000 proteins, only 8 proteins involved in the apoptosis, and all these proteins are not involved in the apoptosis, but these are present in our query protein. So here is a combination like only 9 or in 9 proteins are involved in apoptosis out of 26, and 17 are not involved, but it present in the proteome. And in the query data set, only one protein is aligned with the whole proteome and 13 are not available. So from this matrix, we can make a permutation combination and calculate the p-values. Suppose in apoptosis, less p-value, that more significance. So these are the basic introduction of like Fisher and GCA. Here is a visualization is very important. So like here these are the in our process labs, most of the like enrichment are like depicted by this figure. So I will explain how this figure can be a map downloaded. So suppose like color is just like an enrichment score. So enrichment score like here positive and negative, that's mean positiveness score represent the top and bottom of the list from the ranking, and ball size is define the number of proteins involved in the that particular set of genes. And these are the like all the balls are representing only one pathway. So here is approx 2600 pathways are now existed. So we have to explain in next slide. So it is another visualization. We can visualize our more enriched pathways. So I will also explain how we can do that. So that's the oral introduction. So I now move in like a hands-on part. So everyone should open like Google, like yeah, you can open Google browser, I think. So this slide PPT is like easily understood, everyone. So I can move on next.
Yes, I think. Okay.
Yeah. So I will start with the. So I will start with Fisher. So I'm sharing like our screen for R Studio. Everyone is with.
Yes, we can see.
Yeah. So I will
Yes.
start like first we have to the input file, which is the list of proteins, and just upload these few libraries like Reactome, cluster profiler, or human human on database. So and also provide the Uniprot interest ID, because most of these uh, function works on an interest IDs, not work on like our gene symbol. First we have to add uh, first we have to add gene symbol. First we have to add interest IDs in besides of gene symbol, so they can read easily. Then we go for like using this compare cluster packages and just set the parameter like what the size minimum you want, like minimum to maximum size should be 1500. Set size it is, and p value cutoff you can set, human, all these setting you can make. And then again set the set readable package, they again convert the interest ID into gene symbol, and you can save the file in output file format. And again I'm sharing input file format. Yeah, everyone visible? Yeah. So like this is interest IDs I have added already, and this is the entry Uniprot protein name, and this is gene symbol. So our package is just read this interest IDs only and process accordingly. So we have to add this interest ID in front of all these protein IDs. So this is the our input file format. So can I share these input file format so you can run along with me?
Yes.
Now I'm going to like GSEA. GSEA function. So here also like we provide the input file and just change the interest IDs, Uniprot IDs, and here is the function. So first we have to like arrange the gene ranking and set the all the ranking in decreasing order, so that can read one by one. So this is the function GSEA pathway. So we can set number of permutation. So mainly many like in the literature I have suggested that try to take maximum approx 10,000 number of permutation for better result. So I have just, you can take accordingly your need. And also same thing like p value, min size, maximum size, we adjusted all these values you can set according to you and run same as Fisher. Not too much different, but different is just like this different to provide the ranking in the list. That's, you got my point?
Yes.
Yeah. So I'm also sharing this file. First I show you how it's file format. Yeah, this is like file format for like GSEA. So I have to take read by entry and convert into gene sample and add the interest ID and take this t-test. Yeah, you can select negative login value, login p value. So on the basis of this ranking, you can set the ranking or decrease, run the script. This is very simple. There we can your intensity, your differential, your expression values, anything doesn't matter. So basically, if you don't give this, it is also working. So, but you have to provide two columns. At least first column is the like list of gene names, and second column is the top rank. Do not need for these columns. I have just provided. So basically, if you read only these two columns. Sharing this script. I think you should simultaneously run that these scripts so if you face any problem. So should I move to like visualization? Are you are doing?
On my part, it's fine.
Okay. So I'm starting for visualization. Okay.
Okay.
So first, we have to download the Reactome map where we can map our results, which is as an output file from script. So how we can visualize, like I have shown in the like the PPT, like balls. So first we have to go to download, and here the one option like pathway hierarchical. Go to pathways. Yeah. So here you have to go to pathway, and here select the pathway hierarchical relationship. Okay. So we can download like that. So this is the text file. You can see here is all the organism pathway list. So we have to remove all this. Only keep humans. So it is human. Start with HSA. Yeah, this is HSA. So we have to keep this HSA file that is only for human, and others you can eliminate from the table. So you can this table you can download just like a text file and just rectify that table. Okay, done. Just open the Reactome database and just go to download. In download, go to this pathway and download pathway hierarchical relationship. This file you need to download. Once you download it, just open it, and once you download it, just remove all the unnecessary entries and keep only humans data.
Is it only me, or I don't see anything? Or
No.
did you just share screen? Ah, okay. Just
So
wanted
you
to
have downloaded?
yeah, I just wanted to make sure that's
Yeah, you can remove unnecessary data from that file. Then open Cytoscape. So all that done, Kalin?
yeah. I still can't see the share screen, but
Yeah, I'm not like I'm just opening the Cytoscape because
Okay,
that
okay.
file will be open in Cytoscape. So before opening that file, you have to need the output file. So if possible, you can run Fisher or GSEA. So that file output file generated, then we can use that output file into Cytoscape, mapping over that Reactome database. Okay. Not I'm sharing. I run that. So I'm sharing like Fisher's output. We can map over it.
I think it's on the other screen because what I see in the share is
No,
the
I
download.
I shared output file. So I can go. I'm opening Cytoscape. The Cytoscape is visible. So first open that file which downloaded from like hierarchical relationship file. So I have downloaded this one, Reactome pathway relation. So these are the two columns, HSR, HSA, and HSA. To make it as usual, source node, target nodes. This is over human pathways, all the like 2601 pathways. Here we can see all these pathways information which are linked to each other. So first we have to let's make a very good diagram. So first we have to change the shape and make some customization settings. So go to settings, style. Yeah, screen is visible now.
Yes.
Yeah. So like first go to like to make a good view. So first we have to change. First you have to lock the screen and change the cycle. So and we have to remove this name also. So go to properties and yeah, label transparency column activated. So here is like 255. Make it 0. Okay. So all this are invisible. So like ball size is very small. So we can make a big ball. We can go 3. And edges are very like thin. So we can also make thick edges like here 20. So we can check 25. Yeah, we can also change the color like that. And you can make many more changes accordingly. You can go to the layout tools and just yeah, you can increase width and height also like that. This one like this, no? So like that. Many settings you can do according to your need. So now we have to map our results, which is generated by R script over it by different colors. What we have to do? Go to this table file, import the output file. here is like this ID is key ID, which is common in our pathways. So we have to make this key so that can identified by these nodes. Now all these pathways are assigned, but not showing in screen because we have not made any setting. Just go again in the setting. First go to node. First fill the colors. So fill the colors. We have to select this middle option. So here is many options. So how you can fill color by p value, q value. So we have to just select p value, less p value, less p value, more darker region we can select. So now we have to go continuous marking mapping. Okay. So now we can here see the all these pathway are mapped, which is generated by our R script. So but we have to make more clear. So you can select color. Suppose we don't know what are the p values. So you can set here maximum p value, minimum p value by looking into the table. By the way, it automatically detect the maximum and minimum value. So just you go to and you just make to just change the color because if you here it is default color but it is not represented well. So we have to make a darker color to light color. So we can just visually identify that which nodes are darker. That means these nodes are more significance and more significance pathway representing by particular nodes. So here is many color option you can go, like we have selected red. We can you can all add many more color spectrum by adding this one, like adding one. So you can move down here, make more lighter. Like that. You can add according to your choice. So here you can see, okay, here you can see this node is more significant pathway represented, this less, this medium. So you can adjust color by your like number by according to p values. You can delete it. You can already set like that. So, like you want only more significant, like 0.05 only red. So you can just go and select 0.05. So this parameter is from like up to 0.05. All the nodes will be showing in the red color, and all later all the nodes will be in like other like magenta color and white color. So logic is you can you should understand how you can like change the colors and all these things. So can I repeat? So this is the first setting, and now we can size. So size should be set according to number of set size. Like if you see the table, so number of proteins involved in a particular pathway, so that is called set size. So just go and select like count, number of count. And you can again set the criteria. Like our maximum set size has like 402, so we have to pick a little bit more so we can see the differences like this. Suppose this set size is very like too big, that means that contain number of proteins more. You can check which is the like this is the cell cycle. These set size is involved in the cell cycle. There is border is missing, so we can also add border. Mesh, no? Little bit mesh, so we can order border also. So border we can add according to p value also, yeah, p adjust value. But less p value, darker. You can set any border width. Like that. So here is one more thing, like you are there is overlapping, so we have to remove overlapping. That any option if you're missing a add or remove. Like most of the use we hide and keep it only little like 5 to 6. Also, according to count, we can hide or remove. So here you can see little bit it just go to behind, and like larger cell size is come over all these smaller one. Also you can set from here if you want overlapping or not. So this setting you can adjust from. You can see here that size is very big, so we have to. So these things understood. Because there is too many like red ball, because we didn't we set the p values 1 because 1 is not significant, so we can remove and only select the significant, like 0.25 is enough significant. In our script I had only keep p values 1, so that's why most of the unsignificant path is showing here. You can remove from the note table column, note column is generated by R script. So you want to show only a significant pathway, then you can show here significant. Like in this file, the up to 2.0 0.24 is the maximum p value. So you can set like minimum p values for more significant. So basically, like many papers suggested that for the significant pathways, mostly people recommended 0.05. I have just set the maximum, so all the results are shown. So in the script, you can just set the 0.5, 0.25, any values. If you see my script, then p value cutoff is equal to 1. That's why this.
Sorry, I have a question here regarding the cutoff for the p-value. Is there a rule on how to set it, or is it depending on the type of data that you are dealing with?
No, not depend on. It's like a mostly significant has less p values. Mostly like people just suggest for a significant value you take 0.05 p values. Any the criteria I have for it's on.
So there is no way to fix out how to choose.
Yes,
So
I don't know, but like we never use. So
okay.
use mostly one.
Okay.
So you can use your own p values because it's like too much data, like 5,000, 6,000 protein. Then you can just take 0. Just reduce the set size, just reduce the p value, so you can get the maximum significant values. So here I have like 400 proteins. I have just select one, so we can get the maximum. Maximum
Okay.
Oh, sorry, yeah, screen is.
No, I can share. I can see the screen.
I don't know. Let's see.
These are the basic settings. So now you can save in PDF, EPS, JPG, and any other. Even you can like now if you want to make a more customize these settings, you can have many like attributes, layout. You can go and just set the layout which is most looks good. Any layout as like this version is available. Like in a paid version, Cytoscape, there is a many good version, good templates for visualization. As can use template. There is a limited layout. That is good actually because it is the same. It is represent area is involved cell cycles, area is metabolism. That's why in our live layout visualization, it is easy, so you can just check the which area is like which area is assigned for a particular part. Suppose area is cell cycle, area is apoptosis. These are the basic things. any protein you can select. Select it here. Metabolism, cell cycle. The good format. And other colors you can change according to your any question from this Cytoscape?
No, from my part, it's clear.
I'm now going to like add another visualization. Is online one. Can I go? Then again go to Reactome. Yeah, my browser is visible. Google Chrome is visible, no? Reactome pathway.
Yes.
Yeah,
Yes.
yeah. So for like if you want to just go to take online help to know the pathway enrichment analysis of list of proteins. So just go for analysis tools in Reactome database. These are the layout for this online analysis tools. There is a two option, and other option is also species comparison and tissue distribution, but we need these both two part. First is analyze gene list, so we can paste list of genes, few genes. Is only the continue. These are the results appeared. Cycle is generated. There is a two option. First option is visualization like that. You can download also PPT. That is like the hash format. It's like it because I had shown PPT like that. This is another method. Here most of the proteins. Here this is cell site. I just take random list of proteins. Bigger. Any this is the one method, and another method is like protein with a rank value for that set of. Let's go to like analysis. Here these are the method, but you can choose any one of them. Here we have to provide the data set. Data should be in a two group, like wild type and disease. We have like intensity. Any data, if you see the RNA normalized value, microarray normalized value. Data, I will show you. This data. No. Yeah, this data format is visible.
Yes, we can see.
Yeah, so there is like two groups. Like one group is like male and female. So like this is a female, and it's like male. So there is a two group, male and female. So we have to provide this data. Google Chrome is visible now. Can I go?
No, I cannot see actually.
Oh,
No, I cannot see.
okay. Now it's okay.
Now it's okay. Yes.
Yeah. So let's go for analysis, gene expression, provide go to data, and here if we provide the data file, so we have to make a group. Like this is intensity data. So
Okay.
So we have made two groups. So next, so we have to compare female versus male. Now go to next. No further data on data set. So here is option you can provide the email ID, but all the results can email ID after. And just will take like two larger data set. It took sometimes 15. That is better to provide the email ID. So I can send you once processing complete. Creating like PDF is also Excel file. RScript used in backend. These are the all our results presented. Select any of the can provide more detail for the in this particular pathway. Those detail. Again, we can go for next.
Sorry, can you save this final image in some
Yeah,
format, or if you want to share it with anyone else?
Yeah, I will share. Download PNG. I will share.
Okay.
So it's on you. Give like this. This I will share both of the formats. There are many options like SVG, vector graphics, PNG.
Sorry, this export was one more option.
Yeah, that is one snapshot you can take, or you can take whole image.
And the third one was one more, I suppose. Can you go?
Yeah.
So export, yeah. Export content means that you
all these like SMBL language, but it is not.
Okay. Mm-hmm.
But our
Okay.
raw data we can download from like this. There is option in download.
Okay.
RSP, PDF,
Script. Okay. Mm-hmm. Mm-hmm. Yes. Yes. Yes.
So this is the another format. I miss like up to you format you like most. But here is the problem is that you can't make more like visualization in a good content like I made in the Cytoscape by using different formats of Reactome mapping. Here you can use as it is. You can't make changes here.
Mm-hmm.
So I will recommend you use like script version. So that is better. You can make more clear visualization.
Mm-hmm.
Any question from this part?
From me now. It's all clear.
No, it's interesting. Yes.
So in our next Wednesday, we will discuss about like functional module identification in the protein-protein interaction network.
Okay.
Okay, cool.
Okay.
Thank you for
Share
the
all the
presentation.
like input file. So you can at least try. So all these things will be more clear.
Okay.
So if you found any problem, yeah, you can text me.
Okay. So one more question, may
Yeah,
I?
please, please.
So in that Cytoscape or maybe other, maybe not related maybe to this enrichment analysis, but when you in this software packages, there's some motif to find motifs when you have network protein network. There are some.
Yeah,
You just
sorry.
yeah,
Yeah.
sorry.
Yeah, go on.
Yeah. So you showed us last week, I suppose, that some cluster analysis and so on. So in some option, I suppose that is Cytoscape.
Yeah,
I don't
we
remember.
use Cytoscape and we will use like R script. Like for
Okay.
one method like M code, we can use Cytoscape for like eigenvector, leading eigenvector method, which
Okay.
is used by R. So
Okay. Mhm.
another method which is like Java based, so we can use
Mhm.
this platform.
Yeah. Yes. Okay. Mhm.